Welcome to Beyond Lab Walls, a podcast from the Salk Institute. Join hosts Isabella Davis and Nicole Milnarek on a journey behind the scenes of the renowned Research Institute in San Diego, California. We're taking you inside the lab to hear the latest discoveries in cutting-edge neuroscience, plant biology, cancer, aging, and more. Explore the fascinating world of science while listening to the stories of the brilliant minds behind it. Here at Salk, we're unlocking the secrets of life itself and sharing them beyond lab walls.
Today we welcome Laura Mainz, who's a trained cancer biologist that spends her time at the Salk Institute studying a process of programmed cell death called autophagy in the context of tumor initiation. Welcome to the podcast, Laura. Let's start at the very beginning. Where were you born and raised?
I was born and grew up in Germany, Midwest Germany, close to the border of Belgium and Netherlands, in a very tiny village. We had just 2,000 people, also a lot of farmers around, so probably as much people as cows, I would say.
Was all the farming something that got you thinking about science and animals and plants, or was that anything on your mind when you were younger?
Not really. I mean, I was always curious about the human body, especially when I studied plant biology. Was really not my cup of tea. It was really too boring for me. It was always the human body that I was interested in.
So then at what point did you realize going through school that you might want to pursue science more seriously?
In the beginning, I thought studying medicine would be my path to go. And then when I was in high school, I did 2 internships. My school required 1, but I did 2, one in a hospital and then another one in a pharmaceutical company close by. And I kind of realized that working in a lab is more interesting to me. And that was also the first time that I ever used a pipette. And then I thought, okay, I really would like to study biology. And I applied for a bunch of different universities. And also I really did enjoy biology in school, also math, which many people hate.
So you got to college, you knew what you wanted to do, and it was kind of biology. When did it start becoming more cancer biology?
So when I studied biology, of course, you get the broad introduction and everything. And it pointed out that I was interested in cancer relatively early on. I did focus on molecular biology, human genetics. So these were my 2 majors in my undergraduate. And then why I got into cancer biology, especially in every time into more depth with something, I would say I probably realized maybe retro perspective why I did this. So my father was diagnosed with multiple myeloma when I was in high school. And of course, that was a huge hit for our family. Now I would say I did this, or I did study especially cancer biology, because that's the way I cope with the whole message we got in our family, because I felt very helpless in that moment when we got that diagnosis, and I didn't really know what should I do. And I also didn't really had all the knowledge. I mean, when the doctor told us he has multiple myeloma, I said, what? I never ever have heard about this. And I did the one mistake that you basically actually never should do. I looked it up online. I found Wikipedia and it said 5-year of a survival rate, which was not the case anymore. It was really not up-to-date statement on Wikipedia. And I just felt knowledge helps me in the future to cope with these situations, whatever comes next. He fought 9 years. That's definitely more than what Wikipedia told us. But he was a fighter.
And at that point, did you consider going into like oncology, and were you still thinking about medicine at all, or were you deciding maybe research and development of drugs?
For me, going the academic career was something I never really questioned until I would say last year. I actually started to volunteer for a nonprofit organization here in San Diego. It's called Leukemia Lymphoma Society. And I do patient outreach for them. And also last year, I had the chance to do advocacy for them, especially for pediatric cancer. And that's something I never actually imagined to enjoy, doing patient outreach, working with patients, doing advocacy. So yeah, I'm at the point in my life where I need to figure out if academia or working in an academic field is still what I would like to do in the future, or maybe pivot a little bit more into working more with patients. I can imagine working, for example, in clinical trials would be something that really interests me. But I do really enjoy what I'm doing here right now. These are just thoughts I have for my next step, my next goal.
So how did you end up in San Diego?
I found out about Salk already when I was in my bachelor, in my undergraduate, and I did my bachelor thesis in a lab that had a collaboration with a pharmaceutical company here in San Diego. And the PI in that lab asked or offered me if I would like to do part of my thesis here in San Diego in that company. That was, of course, a huge offer for me as an undergraduate student. So I looked into the company and also into the city and what else is around and found out about Salk. I was really fascinated about that institute, and it just gave me a very positive vibe about how research is done and how good our collaborations are done here. But then I decided not to take the opportunity to go to San Diego because that company also had financial issues. Then a year before I finished my PhD, I again looked into that institute and searched through all the different labs and decided to apply for Jan Karlseder's lab. And then I got here in 2019 for a conference because I just didn't want to do an interview via Zoom. I think in person it's always better. And it was perfect timing because he was looking for someone who already has expertise in autophagy, what I worked on in my PhD.
Can you explain what autophagy is?
Yeah. So to break it down, autophagy is a recycling pathway in our body, meaning, for example, old organelles, old proteins or damaged proteins in our body, which is, let's say, floating around in our body and would probably harm the body because it's not functional anymore, needs to be catched and broken down into individual compartments so that once we have these individual compartments, we can again take them and use them to build new organelles in our body, new proteins that again are functional and healthy and especially young. We need that recycling process to maintain our protein homeostasis in our body. And actually, we can also increase autophagy. I think many people who are interested in fitness have probably already heard it that once you starve your body, you activate autophagy because the body thinks, okay, I need to recycling things in my body to increase my energy, break down things in my body that's not functional, that's old, that's broken. Intermittent fasting, it's exactly that. So basically you starve your body for a specific amount of time, and that induces autophagy and helps to recycle things in your body that usually has detrimental effects if it's just accumulating in our body.
Oh, interesting. That's great. You had the exact specialty Jan was looking for at Salk. Was deciding to leave Germany difficult, or were you expecting to do that after the PhD?
I always had the idea to go abroad at some point in my life. And when I finished my undergraduate, I had the feeling I'm not ready to do this. I would like to stay a little bit longer in Germany. But then 5 years after, it was exact opposite. So I felt, okay, now it's the right time. I'm ready. I would like to do this. I'm just in the right spot to grow into that new challenge.
Do you visit home much, or do you find yourself traveling more in and around North America?
Of course, you always would like to travel more than you in the end do. I have visited a couple of places here, especially in California. Also, before actually coming to San Diego, I visited New York with my family. And then my best friend, she lived in Pensacola, Florida for almost 2 years. So I was there already. And going home probably once per year, I would say, because it's a long flight. And last year, I was very happy because my family came here visiting me for the very first time. And also my little nephew, he is 2.5 years. And he also got the chance to see me here. And I could spend a lot of time with him. That was amazing for me.
Now that you're at Salk, what's your research look like?
So in the lab I'm working in right now, we're generally interested in telomeres. These are the protective DNA caps at the very end of our chromosomes. And we know already through multiple years of studying and based on other labs' work that these telomeres become shorter during aging. And we also know that telomere shortening induces a cascade of different signals, also including 2 main barriers known to prevent that a healthy cell becomes a cancerous cell. And my project is focusing more on the second, later on in life barrier that's called replicative crisis. And it's characterized by an inflammation, but also widespread cell death. And by that, the body basically prevents that aged cells or unstable cells become a cancer cell. So the body just gets rid of them before something worse can actually happen. And then in my project, I established a mouse model where I'd like to understand if this crisis-specific cell death or if defects in this crisis-specific cell death leads to more tumor initiation.
This crisis-specific cell death that Laura's talking about, that's autophagy.
This might be the last barrier before a cell actually becomes a cancer cell. That's the idea of my project. And I've worked on this specific cell death pathway, which is a known pathway in our body that also happens on a normal, regular basis. It's basically a pathway that regulates protein homeostasis. And I've worked on that pathway already in my PhD because it's a known tumor that is deregulated in general in cancer cells, not only in early tumor cells.
You mentioned you're creating a mouse model. Are you mostly working with mouse models of pancreatic cancer, or what type of cancer? Or is it a couple different types?
So in my project, I have 2 different approaches. So my mouse model is focusing on the whole body, meaning I have mice that have shorter telomeres over time, and I combined it with autophagy impairment. And then I see which tumors arises, or do I get one specific kind of tumor type more than others? And where do the tumors arrive? Is it maybe one specific organ that is affected more than others? And then on top of this, I have another mouse model that focuses more on the liver. What has to be known about the liver specifically is that liver cells don't divide very often, while every other cell in our body divides relatively often. So I try to compare if what I do see maybe in my whole body mouse model is the same in the liver, where I have cells that don't divide very often.
Oh, interesting. So you're looking at this whole body model and seeing how shortened telomeres and autophagy can affect cancer development, and if there's a particular area of the body where cancer is more likely to develop.
I don't know yet.
Okay.
I developed these models, and I'm at that point where I have them. It took me 3 years to get there. So it's a long-term experiment, let's say. I'm in that point of my project where I monitor those animals and I will analyze or take the organs whenever I do have a mouse that's sick or it's not going well anymore. And then I take different parts of the body and see if a tumor erases in specific compartments of that body. But I'm not there yet.
Yeah.
If you're enjoying this episode of Beyond Lab Walls, be sure to check out our other channels at salk.edu. There you can join our new exclusive media channel, Salk Streaming, where you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine Inside Salk and join our monthly newsletter to stay up to date on the world within these walls.
Does your lab and your work have any overlap with Christy Tower's lab, since they're also looking at autophagy?
I would say I'm the first in our lab who really works into autophagy itself. So a colleague of mine who discovered the fact that cells in crisis, or in these last barrier, die through autophagy, that's these crisis-specific cell deaths. So he discovered that, and then I continued working in that part of the project. And since we are more interested in tumor initiation and Christy Tower's lab is more interested in how does a tumor cell resist to autophagy inhibition? Because autophagy, again, is a pathway that's deregulated and very often upregulated in cancer cells. So the original idea was, what if we target autophagy in these cells? In cancer cells use autophagy as a recycling pathway to get new energy. And the idea is if we target that pathway, they can't generate energy through autophagy anymore and they die. That was in general a good concept, but of course there are always cells, they are resistant to that, or they activate different pathways to compensate the loss of autophagy. So I think our two labs, or my project and Christy Tower's lab, is interested in two different parts, while we are working more in how is autophagy important that cancer erases. She's more interested in how can we use autophagy as a therapy, and especially can we target it with a second hit once those that has been targeted with autophagy inhibition.
How much overlap are you finding between your research and your findings and research and findings on aging?
Aging is a cancer-related disease, so one comes with the other, I would say. Before I came to Salk, I was not really interested in death in any age-related disease. It was always understanding cancer and trying to understand what has to go wrong, because there are so many processes in our body that just go smooth. There's nothing happen, or at least the body's always able to cope any, let's say, mistakes. But what has to go wrong that a healthy cell actually becomes a cancer cell? That's super interesting to me.
Do you have any questions that you're excited to ask in the future?
Asking questions, it's our daily business. The main question is really, why is this specific process, or can this specific cell death really be the last barrier, or is there maybe something afterwards where the body implemented maybe a third barrier that we don't know about it, that again prevents the transformation of a healthy to a cancer cell?
And is there anything else that you do other than lab work?
I do really enjoy having a work-life balance. So I do enjoy doing my work here, but we dedicate a lot of time and hours to our job. So I think I like to have a clear cut sometimes of, okay, I try to switch off my brain and just go home and enjoy other things than science.
What are some of your favorite things to do when you're turning off your brain at the end of the day?
In general, I like to be active. outside of the lab. So I'm a runner. I go to the gym. Fitness in general, I do really enjoy going for a hike with friends. In general, activities with friends, I do really enjoy. I'm a dancer. So I started ballet when I was 7. Did a bunch of different types. So I did ballet, jazz, hip hop, modern. Then I got lost track a little bit. And then once I actually came here and the pandemic was a little bit over, I started to salsa, Cuban salsa, bachata. So I got back into it. That's something I really do enjoy. And yeah, since last year, the volunteering work, helping others was always part of my life.
So can you still do all the ballet positions?
I can. I think that's something you never forget. Yeah, just the split. That's a difficult thing.
I was going to ask with the dancing too, do you feel like that kind of creative hobby outlet has felt totally separate from the science in your life? Or do you feel like that kind of creativity throughout your life has inspired or helped you to ask more interesting questions or to, think outside of the box more in the lab?
That's a difficult question, and I never thought about that. I mean, I have to say I stopped with ballet in the same year when I finished high school and started my undergrad. And I never really considered myself as a creative person, but dancing is kind of being creative, but just with your body, not with other skills. I think for me, dancing and being active, it helps me to switch my brain off, actually. So probably the opposite of what you just thought, because again, we spend so much time in thinking about science and problems and things we like to solve. It's good. We have our moments where we actually say, okay, I'm not thinking at all.
I think it's wonderful to have the work-life balance. It's very important, especially when you're doing such hard work and you're doing something also that is so fulfilling to you. And it feels like you're able to target this feeling of being out of control, put it to work, and use that as fuel in the lab. What's exciting you most about your research in the years to come now?
And I think for me personally, nowadays we are all focused on finding a treatment for cancer. And don't get me wrong, that's very important. But I think we sometimes forget that we also should focus on the fact that maybe we should take care more of our body and make sure that we don't even get in the first place. And the reason why I mentioned this is that my project focuses exactly on that. So what do we need to know about cancer tumor initiation that helps us to prevent cancer in the first place? Because just hearing the words, you have cancer, and that was not even to me, that was to my father, has such a huge impact on you and your life. And I mean, in my family, it was a cancer that is treatable, but it's not curable. And even if it's a cancer that's curable, it still affects you a lot. And I hope that in the future, less people actually hear these words.
Well, thank you so much for coming and talking to me and sharing your story and your science.
Thank you. Thanks for having me.
Beyond Lab Walls is a production of the Salk Office of Communications. To hear the latest science stories coming out of Salk, subscribe to our podcast and visit salk.edu to join our new exclusive media channel, Salk Streaming. There, you'll find interviews with our scientists, videos on our recent studies, and public lectures by our world-renowned professors. You can also explore our award-winning magazine, Inside Salk, and join our monthly newsletter to stay up to date on the world within these walls.